ViruSurf is a novel platform to search for virus sequences in a flexible way using both their describing metadata and their variants. The pandemic outbreak of the coronavirus disease COVID-19 caused by the virus species SARS-CoV-2 has created unprecedented attention towards the genetic mechanisms of viruses. The sudden outbreak has also shown that the research community is generally unprepared to face pandemic crises in a number of aspects, including well-organized databases and search systems. We respond to such urgent need by means of a novel repository and search system collecting virus sequences and their properties, so as to facilitate current and future research studies. As to our background, on the left we see a conceptual model proposed for human genomics data. In the center we see the pipeline implemented for genomic data integration, and on the right the ViruSurf web interface for querying the database of genomic signals. Thanks to such previous knowledge in human genomics, we have been able to rapidly design and deploy ViruSurf. We recently applied our expertise in data management to the wide landscape of virus-dedicated resources to be able to provide a novel system. Our schema is general and applies to any virus. The sequence of the virus is the central information. Sequences are analyzed from the biological perspective, such as virus species and host environment, the technological perspective, like sequencing technology, the organizational perspective, which is a project responsible for producing the sequence, analytical perspective, including properties of the sequence, such as known annotation and variants. We integrate public data from different DNA or RNA sequences with their annotation. We enrich it with variation data, such as mutations, computed with a sequence alignment algorithm. In ViruSurf, we include SARS-CoV-2 data from GenBank, CogUK, and MDC. We also include SARS-CoV, MERS-CoV, Ebola, and Dengue sequences. With GISAID, we have a special agreement, and we can include them in a different interface, ViruSurf GISAID. The interface is composed of 4 sections: a menu bar to access the different services, documentation, and query utilities, the search interface over the metadata attributes, the search interface over annotation and nucleotide amino acid variant information, a result visualization section showing a flexible table with the resulting sequences described by their metadata. The interface enables an interplay between search performed within parts 2 and 3, thereby allowing to build complex queries given as logical conjunctions of arbitrary length of filters set in 2 and in 3. We perform extensive interviews to groups of virologists of various specializations. Each researcher has provided us with a viewpoint on applications of virology that serve as requirements for progressively adding relevant features to our database, as well as relevant search services to comply with their needs. We expected our future focus to be on users researching on diagnosis, working on vaccine development, or drug resistance. Existing systems typically offer very nice visual interfaces reporting results of data analysis but limited search capabilities. By means of complex search queries over our database, it is possible to help virus research according to the requirements provided by several domain experts. We really hope you will like our system ViruSurf. You find other videos that explain practical queries of example. Try for yourself!